Thank you for the introduction. And my name is Tetsuya Yagi. Actually, I started my research career with retinal physiology, and I'm still working on the primary vision system, and I'm a physiologist. And back to 1995, I think, I happened to have a chance to attend the Neuromorphic workshop in Telluride, where I met Toby and Misha and those nice people. And I was very interested and fascinated to see the neuromorphic vision system, but not necessarily because to intending to design the fancy cutting-edge vision system, but rather I want neuromorphic system which is able to reproduce the response. Oh, battery. Well, I was interested in neuromorphic vision system, which will be able to reproduce a response of retinal neurons in real time. So that's my motivation. And I first want to make an acknowledgement because I'm afraid I am not able to finish my talk in a given time. And I can just speak slowly, and if you make me hurry, maybe my talk turned out to be in Japanese. So this is those people contributed the research I'm talking today. And basically, the research has been conducted in my lab from early 2000 to 2021. And some of the people here moved to, promoted to the other universities and to run their own lab. And those people, among those people, Professor Hayashida now and Professor in Mie University contributed a lot. And here, Professor Weiji Song, he joined my lab, my group, in very short time. But he's a specialist of the voltage-sensitive dye imaging. I'm going to show it today. And he instructed us to make such a fancy imaging technique. And last but not least, I want to thank to the master course students in my lab. And unfortunately, they contribute a lot, but unfortunately, only few students went on to a PhD course. And nice students prefer business rather than academia. So that's too bad, but we have to think about that situation. Okay, so let me start with the basic physiology of the retina. And as you can see, the retina resides at the bottom of the eye. And retina consists of 5 major types of neurons, like photoreceptor, horizontal cell, bipolar cell, amacrine cell, ganglion cell. And retina is actually a part of the brain in terms of developmental aspect. And here, what's going on in the retina, and photoreceptor here transduce the light into the electrical signal. And that signals are processed in the circuit, retinal circuit, and out of neurons are ganglion cells. And I want to emphasize here, most of the retinal neurons, except for ganglion cell, respond to light with graded potential, slow graded potential, not give rise to action potential. And ganglion cells are neuron, only neuron, some amacrine cells and ganglion cells, they produce action potentials to send the signal to the brain. That's the very important point. Something like this. And then this is a light evoked response, or actually photocurrent. And the photoreceptor. Here, flash of light induce the change of photocurrent, this slow time course. And as we increase the intensity of light, the amplitude goes up and saturate. So I want to again emphasize that time course is very slow. The peaks at like 100 milliseconds or so. But it's depend on the adaptation level. Okay, so as you know, the neuron function of the neuron is characterized with so-called receptive field. So concerning receptive field, the center surround organization is fundamental, and that is calculated with the first neuronal network in our visual system, consisting of photoreceptor, bipolar cell, and horizontal cell. And the bipolar cell takes the difference between the response of photoreceptor and horizontal cell. And here, the important structure is so-called syncytial structure in biological term, and which is the neighboring photoreceptors are coupled weakly, electrically, and neighboring horizontal cells are tightly coupled electrically. So then what happens is that the response spreads to neighbors, and in case the photoreceptor, the voltage response spread a little bit to neighbors, and the horizontal cell diffuse almost over the entire retina. And here represent the, say, point spreading function, and photoreceptor layer and horizontal cell layer, and the bipolar cell takes the difference between these two, and give rise to this kind of center surround organization. It's a very important point to understand the function of the retina. And a little bit more about the mechanism of this center surround suppression here is, as Toby introduced, the horizontal cell can be modeled with the resistive network, rather kind of resistances here. And simple calculation gives us that the point spreading function here in one-dimensional case goes off with the exponential manner. So the exponentially response decay. And which means that the two signals of the photoreceptor crosstalk each other in this layer. In other words, two signals generated in the photoreceptor correlates somehow. And correlation goes off exponentially, quickly, with exponential function. And this model explains very well the real retinal, physiological experiment. And this is quite very remarkable. And this model first proposed by Ken Naka and William Rushton, a very famous physiologist. This good. But it is more interesting to think about the function of the retina from this nice paper published from David Field. And he took a picture of natural scenes like trees and rock and whatever. And he analyzed the power spectrum of the spatial frequency component. And he found that more or less the spectrum can be fitted with the kind of first order model. It is like power spectrum goes off with a power of 2, so minus 2. It's like a RC circuit in time domain. And that means that in the image, arbitral 2 points are correlated to each other. And correlation goes off as a distance. Okay? So, and actually exponentially. So what this structure means is that the horizontal cell remove the redundancy included in the image. So this is a very important, I think, interesting and very important interpretation of the function of suppression from horizontal cell. So the retinal circuit is well adapted to process the image. That's I want to emphasize. Let's go on to the next one. And this is ganglion cell that I wanted to show with a sound, but no sound. I'm sorry. And here is the light. Here it represents light. And in this case, ganglion cell, this ganglion cell gives rise to a high frequency response after light on. And different one here. Another one here respond well to, Yes. Go back. Here go. Here, another one. Where is respond to that cessation of light turning off. So this one is called on type and this one off type. And a little bit more about ganglion cell. And we record the response with this setup, physiological setup. And here the retina is isolated from a mouse and put it on a dish on the multi-flat electrode. And the retina is superfused with oxygen superfusate. And we give stimulation through optical pathway of the microscope. And sometimes we stimulate with spot of light or annulus of light or checkerboard kind of image. And even the image like this to see what's going on in the ganglion cell like this. And those are some examples of ganglion cell. And in this case, so light, this represents the period of light on and light off. And in this case, this cell responds well to the light off period. And here, light on, and we are able to find several different kind of response patterns in ganglion cell. But basically, those ganglion cell response consist of transient part and sustained part. And that was explained by Toby in the previous talk. And how we can characterize the receptive field or temporal receptive field. And we'll characterize a receptive field with a so-called spike-triggered average. Spike-triggered average. We apply randomly modulated light and get spiky response. And we cut out the stimulation with some beam every time we find the spike. And we sum up those stimulations and to get this kind of profile. And this means that the intensity profile this intensity profile the most probable to induce a spiky response. So in other words, this one is the temporal receptive, so to say, temporal receptive field defined with this experiment. And when we analyze the frequency component of this temporal response, temporal receptive field, the frequency component is something like this, and basically bandpass, because biphasic feature. And this one actually, so the ganglion cell, so-called off-alpha ganglion cell in the mouse. And the power goes off very quickly at the 10 hertz or something like that. So again, the temporal receptive field is kind of has a slow structure. Okay, so that is very important to understand the retinal function. In other words, those slow receptive field plays important role to filter the incoming image in real time. That's another important message I want to emphasize. Then, based on those physiological observations, we try to build the neuromorphic system, which is able to reproduce the response of the ganglion cell. And those are the requirements of our neuromorphic retina model for in vivo experiment. I mean, it is impossible to record the bunch of ganglion cells at the same time with physiological experiment. And also the eye movement is a very important factor to understand the function of the retinal visual system. So then the first priority to build the neuromorphic retina model is the compact hardware to be mounted on the robot eye because of the eye movement. If the system is compact, we can put it on the robot eye to make it move around that. And second one, reproduce the receptive field, but within the resolution of graded response. So as I showed that the graded response is quite slow, and the frequency component included in the response is kind of low. So the photoreceptor of the neuromorphic retina is not necessarily the continuous sampling. So far as sampling frequency is high enough to reproduce the photoresponse and temporal receptive field, that's okay for me. And the programmable architecture, this is to emulate the different kind of ganglion cells. Ganglion cell has several 10 classes, subclasses, or something like that. And we need to duplicate that receptive field by changing the parameters of the neuromorphic retina. And real-time computation, this is necessary. So all the computation can be, should be done in real time. Okay, and as a secondary priority, low power. But not necessarily super low, like fancy vision system introduced by developed by Toby. And continuous sampling, that's a second priority. But should be fast enough. And fully analog, it is not also not the first priority because it should be programmable, easy to access to digital technology. That's more convenient to work with. And resolution is not the first priority, and those kind of things. And based on those requirements, go back to 2003, we developed the first version of the retina, neuromorphic retina, and that is introduced by, briefly introduced by Toby. And just briefly mention, I want to mention the two resistive network representing photoreceptor layer and horizontal cell layer. And the bipolar cell takes difference between two. And there are switches and memories in the circuit. And I want to mention here's a switch. And this operates in such a way that during the circuit is taking difference between the two resistive network to generate the center surround organization, this switch off. And during that period of time, the photosensor accumulates the next frame. So basically, the computation is continuously conducted in frame base. Okay. And this is the output. The horizontal receptor layer, horizontal cell layer, and the bipolar cell layer. And oh, I forgot to tell you the resistance of this. Oh, this way. Resistance here is transduced actually, so we are able to control the resistance of this resistor. So we are able to change or control the receptive field, wide one or narrow one, or a sharp one, or something like that. That's an advantage. And here is the different resolution here. We changed the resistance, connecting the neighboring pixels. And using the analog using the analog memories, we are able to get the frame subtraction, something like this. And here on type responding to light on, and this one is on off type, something like that. And let me show you some application. And since we are able to compute the spatial, or compute with the spatial receptive field when the photosensor is accumulating next frame. And during that period of time, we are able to change the receptive field size in time-sharing manner. And this one here, 3 resolutions, fine to coarse images are obtained within one single frame sampling time. And this is one interesting application, I think. And this is another application, and this is the collision avoiding eye system. And this tells the time to collision with one eye. And actually, this one is inspired by the collision avoidance neuron and insect or some kind of birds. And the algorithm is something like if we approach the object with the constant velocity, the object gradually, image of the object gradually increase, right, expand. But as we get closer, the image expands exponentially. So we use the image change to predict the time to collision and make this small vehicle to stop, something like that. This is another application. But the first version does not include temporal property, the temporal receptive field. So we calculated the temporal receptive field with FPGA. So since the temporal receptive field is kind of consists of very slow component, free-casing component, and that one is not quite easy to implement with the analog circuit. So then we did that with FPGA, and we have the photoreceptor, horizontal, and the horizontal suppression is somehow delayed with the center signal, and take difference and then calculate with the temporal receptive field implemented with the IIR filter, combination of the IIR filter, infinite impulse response. Okay, and then nonlinear rectification and spike. And spike was actually calculated with the by solving Iskevich equation, something like this. So power is not low. We require some power, but that's okay for our purpose. And this is to show the difference between the first version and second version. And now the difference is quite clear that left, no dynamics, no temporal receptive field, and right one, temporal receptive field. The big difference is, as you can see, this is slow motion. In every frame, this waving hand occlude the face behind. But in case of with dynamics, the waving hand does not disturb to look at this face, and moving object is removed. And this happens in your vision, actually. And if you look at someone's face and waving hand, and if you wave hand very slowly, it occludes. But first case, this doesn't disturb. This is very effective. You have to gaze some point. Otherwise, you follow the moving hand. So this is not a case. You have to gaze at some point or somebody's face, and yeah. And this is quite effective temporal filter. And the other way around, and this mimics the eye movement, smooth pursuit. And with dynamics, and right here is with dynamics. Temporal, with temporal receptive field. As you can see, the background diminishes, and it looks like the object is popping up. And this is also the case in case of our vision, like something like that. This time you have to follow the object. Okay. And those demonstrated that the effective filtering in retinal neural circuit actually simulated with our neuromorphic vision system. And we, using our second version, and we try to duplicate or reproduce the response of ganglion cell. And this is the response of alpha ganglion cell. And stimulation is this randomly modulated one. And as you can see, the ganglion cell gives rise to response here, something like that. But it looks kind of intermittent. So the this pose. So it looks like the ganglion cell is derived with some event. We don't know. hopefully the event which is useful or meaningful to our vision. So the optimistic interpretation of this is even in the retinal cell can pick up the event which is meaningful for our vision. That's optimistic interpretation of us. Okay, and this is the simulated response with our neuromorphic retina. Spike frequency is a little bit low, but basically the phasic response are intermittent, and it's quite similar, I would say. Okay, so now we are ready to work on the so-called the virtual in vivo experiment, and we with the eye movement. And I want to tell you some background in the retinal research, and the function of the retina or response is interpreted with, has been interpreted with the rate coding. That one is the averaged response frequency in some bin encodes the information. But recent physiological experiment and suggests that the retina function is more than that. And if we go back to this, the number of spikes along this time is kind of quite low. And so that means that the frequency here is 200 to 300 hertz, but very transiently. But usually response is, say, 10 hertz or something like that. I mean, the spike frequency is 10 hertz or something like that. So that means that suggests something like a timing code. So the very small number of spikes encode some information, ganglion cell. And those are the new interpretation of the function of the retinal neurons. And we wanted to figure out what information is encoded with small number of spikes. So to do that, our silicon neuromorphic silicon retina, I believe it's very useful. And this is a simulation here. And the response to moving bar in front of this. And this one is slow motion. Okay, now bar. This is a bar, stop and go.
Can you play it again?
Oh yeah. Slow one, eh?
Yeah, slow one.
Not straight, right? So actually, I don't know, but if you move some bar in front of some object, maybe at the border, we have illusion. Yeah, I think so. We have vision, so that's okay. So, and here mimics the eye movement, saccadic eye movement, and response is something like this. Each dot represents a ganglion cell spike. And we decoded the output with ISI. ISI is a spike interval. So we obtained this image, decoded image, and with rate code within 20 milliseconds or something like that. And this is quite different from this one. And we did also the next one. This mimics the microsaccade. So even the time we are gazing at point, our eye has small saccadic microsaccade eye movement or micro movement of the eye, and so we duplicated or reproduced the situation with our neuromorphic silicon retina. So I hope those can demonstrate that how we can use our neuromorphic silicon retina to understand the function of the retinal circuit. And we are now working on this, and I'm not going to talk much about this. Okay. So this is first part of my talk. If you have any question, happy to. Is that okay?
Yep.
Okay, so here we stimulated the isolated retina with this image. And in this case, actually what we are doing is that we get the recording from the single one ganglion cell, and we move around this image to see how that ganglion cell respond to the different part of the image, something like that. So the message I want to say is actually this sparse spike density again. So in case of the randomly modulated stimulation, we targeted only a central part of the receptive field. I mean, a central excitatory receptive field. And in this case, it's image. So the targeted the stimulation to the center of the ganglion cell. It doesn't happen. It doesn't happen in everyday life. But usually the image is important. So in that case, because of the mixture between the excitatory region and inhibitory region, the response gets farther less. So very small number. And this actually rings a bell. And 2003 or 4, I joined the video prosthesis project. And that video prosthesis project is the retinal implant, actually. And I was working on that. And I tried to design the electrical circuit. And one of the big problems is the power dissipation in this case. Here is the image processing unit, and then the external processing unit, and then embedded one internal module. And this is under skull. And this drives the multi-electrode implanted onto the video cortex. So the serious one is, of course, internal module. And this has to be low power. Otherwise, our brain burns down. So the stimulation, even though the southern channel, stimulation is no problem. So we stimulate the tissue just very short time. So it doesn't require much power. But problem is its communication between external and internal module. And here is some tip to develop the low power, the internal module, low power communication between external and internal nerve module, and the low density of the spikes, and which greatly reduce the communication rate, and then low power. And that is the one, I think, important application of our neuromorphic retina. And let me explain a little bit about the history of the visual prosthesis. And it has been well known that the stimulating the visual cortex provoke the small, confined light sensation called phosphene. And those people studied the characteristics of the phosphene intensively and using the volunteer patient. And patient report to actually when they stimulate the visual cortex and patient see or feel the phosphene at the location in the visual field predicted with the visuotopic mapping, roughly speaking, very roughly speaking. So, which means that if we stimulate the visual cortex corresponding to the foveal vision, we see the small spot of light in foveal receptive field, foveal visual field. Okay, but very roughly. And experiment is, of course, the verbal report. And we ask patient to put the kind of dot where he or she could see the phosphene or something like that. And sometimes phosphene is large, sometimes phosphene is small, sometimes phosphene is surrounded with halo or something like that. So quite unpredictable. So we thought that it is necessary to find the relation between this phosphene sensation and activity induced excitable activity of the visual cortex. And we did experiment using the photoreceptor imaging. And this is something controversial idea concerning the activation of the visual cortex. And conventionally, many people think that, accepted that the electrical stimulation excites some very confined area around the electrode. But those people made the nice experiment with the confocal microscope and find that they suggested that the electrical stimulation stimulate or excite mainly the passing fiber in the tissue. So, which means that excited cells are spreading around, like looking at a star or something like that. This is not this is not good for visual prosthesis. Visual prosthesis, the confined phosphene has to be evoked or provoked. Okay, so then we conducted the experiment, something like that. We stained the brain with photosensitive dye of mouse, actually, and photosensitive dye is very good dye, and which, in a sense, that photosensitive dye is able to pick up the voltage, membrane voltage itself, so quite fast to respond, different from the calcium imaging or something like that. In case of calcium imaging, the fluorescence change with the calcium, the concentration, so kind of delay. But photosensitive dye directly measures the membrane potential. But problem is the signal is very small. So photosensitive dye, the absorption spectrum or fluorescence spectrum changes depending on the membrane voltage. So, but problem is the signal is very small compared with the background illumination or background fluorescent or background the absorption. But elaborating the setup, we were able to record the excitation induced with the electrical stimulation. This part is the V1, and the surroundings are higher order video area. And when we stimulate here, the excited region spread to the second order, or maybe area corresponding to V2, V3, or something like that. So this is good. And something that bothered us is, maybe you noticed that the excited area spreads all over V1. This is not something required for via prosthesis. It's like giving a shock and then white out. So this bothers me a lot. But in 1920, very recently, the Israel group also showed the electrical shock induced the spreading response, excite region. So I was kind of embarrassed to see those. Embarrassed is not a good word, but I was confused. But I found that, this is a difference of the stimulation sequence used in clinical experiment and our experiment. And in case of clinical experiment, they use the repetitive pulse. And in my case, the single pulse. And Israel group, 3 pulses. And we applied the repetitive pulse and something. Let's forget about those parts. This is. And soon after the electrical stimulation and signal spreads, and excitation spreads. But soon it gets small, and settle down within 4 or 5 spikes. So I believe that this area corresponds to phosphine, and maybe spreading signal somehow represent the halo. Sometimes patients see surrounding halo, halo or some that patients see. So it's a nice correspondence between the excitation and, I believe, the sensation. So we went on to find out what is the necessary intensity of the current using the voltage sensitive dye. And this is actually the charge required to excite the cell. And this is response amplitude, normalized response peak. And as you can see, we get this. And quite good match with this equation. And here, 10, I think this corresponds to 10 microamp. Yeah, 20 microamp of current. And this is acceptable. And some people misunderstand that okay, we have a very fine and nanotech electrodes, and we can apply this to get the high resolution of the phosphine pattern. But actually, as the electrode gets smaller, the resistance increase, impedance increase. So say 1 mega ohm of the impedance, we have to apply 10 volt, 1 mega ohm in the case, 10 microamp to get the 10 microamp we have to apply 10 volt to get the 10 microamp stimulation. If 10 mega ohm, we have to apply 100 voltage, it's disaster. It's a disaster. So those range is acceptable limit to excite the cortical neuron. And we encouraged with those observations, we developed a multi-channel stimulator chip or something like that. I'm not talking much about that, but I want to encourage you to find the feasibility of the visual prosthesis with this slide. And this simulation is a simple case that the phosphene is regularly arranged. This is impossible to do that, I know. But suppose that this phosphene is arranged in a square array or something like that. And here is the response. This is simulated phosphene. Okay, something passed in front. And actually, the input image is this. And feasible phosphene pattern is something like this. Phosphene is somehow blurred. And some phosphene is large, some phosphene is small. And in that case, what we obtain is this. Not bad. There's no difference between. And even this case helps more, I would say. And another simulation here is here. This was calculated, the location of the phosphene was calculated using the physiological map here. And actually, we simulated the case that the electrodes are arranged in regular spacing. And in that case, the phosphene pattern could be like this. Just if we calculate with the physiological map, inverse physiological map.
Can you play that one again?
Sorry?
Can you play that video again?
All right, yeah. So, how to find the vehicle is passing in front in this case. But this is more feasible phosphene pattern. So even though we regularly arrange the electrode, phosphene have a jitter in location, this and that, this and that. We simulated that case with this. So this one is much better, I would say. So this regular arrangement disturbs our finding the situation, right? So this also encourage, I would say, to work on visual prosthesis. And here the simulation. And we are conducting the kind of psychophysical experiment with head-mounted display, something like that. And many people think that the restored vision should be something like a blurred image or something like that. But actually, that's not the case. What we restore in our restore is the phosphene pattern consists of the dots, 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 dots. But this is a biological motion. We have a very sparse spot that even though we are able to tell that this person is here, person is dancing and walking, and even here, maybe this person is laughing or something like that. So the restored vision is something like this. So we have to understand this to go on the visual processes. If you expect the kind of restored vision like what we see, it's quite different. So based on this, accepting this, I think we are quite feasible to the visual process is a kind of feasible technique to restore vision. And we are working on this circuits, and I don't want to talk about that. This final message is here is retina communicate to brain with very sparse number of spikes, and yet the brain is able to interpret the visual situation, ambient visual visual situation, and we are able to maybe utilize that communication and processing and communication mechanism for the visual processes as well as maybe AI and this kind of thing. So this is my final message. So thank you very much.